Welcome everybody. Thanks for coming today, joining us in person, or if you're there, on Zoom or in the live stream for the thesis defense of Michaela Melanson. I'm Scott Hamilton. I'm her thesis advisor, and I also want to thank her thesis committee who's here. So Wes Heim and Max Grand, thank you. And then some of Michaela's family was able to join us as well. So her mom Maria, her dad Michael, and her boyfriend Daryl. Is that right? Yep. Right. Great to meet you guys today. And so I'll do a little introduction. Michaela will tell you then what she's been doing for the last 3 years for her thesis. And afterwards, we'll have some questions. Then her committee, we will take her away for a few minutes to talk to her a bit more, and then come back and the celebration will be on. Right. So a little bit of an introduction to Michaela. So I got a lot of nice photos from her parents. Right. There was some early signs there that she would, excel in this sort of academic career. Right, in all of her academic pursuits, right? And that she would probably be focusing there on the marine and aquatic sciences. She looks very intent there with what she's doing. Before coming to see us here at Moss Landing Marine Labs, Michaela did her undergraduate degree at UC San Diego, so she graduated in 2020, and great grades and a lot of really good research and other experiences. So here's a few highlights. So one of the labs she worked in was the Center for Microbiome Innovation, where she studied the effects of different aquaculture practices on bluefin tuna and their microbiome and how it affected their microbiome. And she was even the third author there on a publication, so way to go. She also spent a summer as an intern for the California Collaborative Fisheries Research Program, which is the statewide marine protected area monitoring program that I run here at Moss Landing Marine Labs, but she was working down there for Bryce Simmons, who leads the group there in San Diego. So got to go out fishing, catching and handling lots of fish. And then she also spent some time working with one of my good buddies, Stuart Sandin, in his lab there at Scripps. The project he runs called the 100 Island Challenge, where it's studying the coral reef ecology of 100 different islands plus throughout the Central Pacific. So she had a whole wide variety of experiences before coming here. I learned very quickly that Michaela is very intelligent, strong, she's a good competitor, she's very determined, she's very focused. Right, and she's multi-talented. I mean, she's finishing her master's degree here in 3 years, which at Moss Landing is quite an accomplishment, especially during the COVID years. Like, how did you finish it so quickly? It's really impressive. Whenever I would meet with her and make suggestions, she would come back like a week later, having done that plus 20 other things. Her thesis is like almost 200 pages long. It's really impressive just kind of how hard she worked. Very few students, when they set goals, they actually meet those goals, and she would meet those goals and exceed them. She's like, I'm going to analyze all my gut contents in 1 month. I'm like, no way, you're not going to do that. You're going to be so bored looking at fish guts. And she did it. I was like, how does she do it all? Amazing. But along the way, she maybe passed up a few other potential careers. Right here, she could have been a competitive eater. You could have won the Nathan's hot dog contest, maybe. Right, but you chose marine science instead. She was a very talented dancer and accomplished artist there. I mean, if I put my body in those poses, it would be broken permanently. Like, how do you do it all? Impressive. But really, there was something about the ocean, I think, that really captivated Michaela, and she decided to pursue marine science. And so here's a poem that her dad sent me. He said really sort of encapsulated who Michaela was. This is an excerpt of the poem. It says, the sea, the open sea, the blue, the fresh, the ever free, without a mark, without a bound, it runneth the earth's wide regions round. It plays with the clouds, it mocks the skies, or like a cradled creature lies. I'm on the sea, I am where I would ever be, with the blue above and the blue below, and silence wheresoever I go. If a storm should come and awake the deep, what matter? I shall ride and sleep. I love, oh, how I love to ride, on the fierce, foaming, bursting tide. With every mad wave drowns the moon, or whistles aloft his tempest tune, and tells how goeth the world below, and why the southwest blasts do blow. I was never on the dull, tame shore, I feel the same way, but I loved the great sea more and more, and backward flew to her billowy breast, like a bird that seeketh its mother's nest, and a mother she was and is to me, for I was born on the open sea. So, yeah, I mean, I pretty much 2/3 of all the pictures I got was Michaela on the beach, in a lake, in the ocean, somewhere. And so she's had a lot of great experiences. Right? She also, her parents, have been very supportive, right, and instilled in her, kind of the scientific ethos, right? And so here's another quote that her dad sent me. saying, passionate concern is not a substitute for rational analysis and a rigorous application of the scientific method. And for sure, I mean, Michaela has always been that very objective scientist, despite you can see, there's some passionate concern happening here, right, in some of these photos. But, she really did, believe in applying that scientific method in a very rigorous and objective way for her thesis, which she'll be talking to you about shortly. So she applied that ethos very well to studying the trophic ecology and mercury concentrations of Canary Rockfish along the west coast. It was part of a large project that we did, sampling them from Washington to Southern California, funded by NOAA. It was trying to understand kind of what's happened to this fish species. They were really heavily overfished and then rebounded, and what was kind of their current status. And so Michaela played a large part in that project. There are a few other kind of, academic highlights for her time here beyond the Canary Rockfish project. So she was also employed for about 6 months by the Nature Conservancy, where they were really interested in learning more about fisheries for sharks and rays and potential bycatch issues. They were hoping to kind of move into that space. And so Michaela did this huge, extensive literature review, put together this big slide deck and this report for them on kind of the current status of elasmobranch fisheries. She was also awarded a summer internship through CSU Coast at the NOAA Long Beach office, where she was working on highland migratory species issues, and she was working on that species, the striped marlin. And again, this was all during COVID, so it was all kind of remote stuff, but she did a ton of assessments of kind of, looking through all the stock assessments, what data was available, and compiling all this technical information into a big white paper and a big report for them on kind of the conservation status of striped marlin. She was also awarded a scholarship here at Moss Landing, the Sonia Linnick Hamilton Marine Science Scholarship. Right, also looking through the photos her parents sent me, this was a screenshot from her computer, I think when she graduated college at UC San Diego. Thanks, Mom and Dad. I couldn't have done it without you, without your never-ending love and support. I can't wait to make you proud and swim with the big fish during my Master's in Marine Science at Moss Landing, which I think you will see she has definitely done, making all of us proud by this hard work. So with that, congratulations, Michaela. You're in there somewhere. Right. All right, let's hear from her what she's been up to the past couple of years.
Perfect.
Right, away you go.
Thanks. Okay. So Scott kind of mentioned this already, but for anybody who has not been aware of what I've been working on the last 3 years, the title of my thesis defense is the Trophic Ecology and Mercury Concentrations of the Canary Rockfish in the California Current System. So, in order to talk about a trophic ecology study, we must also understand the structure and function of marine food webs. Marine food webs in design are pretty modular. They have a really broad base of the food web that comprise of marine plants such as seaweeds, as well as phytoplankton and zooplankton. And then as we move up throughout the marine food webs, we have our mid-trophic level species. Those are our primary herbivores, our primary carnivores such as fish, vertebrates, squid, etc. And then we get to the top. The apex predators of the marine food web tend to be a bit lower in abundance, but still biodiverse. We have our large predatory fish, we have our sharks, our marine mammals, and our marine birds. And it's important as we look through this marine food web that the mid-trophic level of these marine food webs act as a funnel and a bottleneck sort of energy transfer from this broad basal resources that we have at the bottom, tends to be both biodiverse and highly abundant, till we get to the top, which seems to be less abundant but still biodiverse. The mid-trophic level acts as sort of a bottleneck for energy transfer from the base to the bottom. And this is performed through predator-prey interactions. Predators forage and eat very differently throughout the marine food web. We have generalists, specialists, opportunists, as well as parasitism and other specialty foraging strategies such as that. But an example here, we have the summer flounder. This would be identified as something as sort of a generalist. It has a wide variety of prey items versus something like the Atlantic croaker. This is more of a specialist. It specializes in one or two types of prey items. And this is important because any sort of natural or anthropogenic perturbation to the system can impact whether or not that species is able to survive. For example, if a natural fluctuation to the environment was wiped out mantis shrimp and sand shrimp, In this environment, summer flounder would still likely be able to perpetuate in its range just because it can switch its resource allocation to things such as fish as opposed to the shrimp. Whereas something as the Atlantic croaker, if there was a natural perturbation to the system that wiped out worms from the system, that natural perturbation might not necessarily impact the abundance of Atlantic croaker, but since its trophic resource is eliminated from the system, it would likely no longer be able to perpetuate throughout that system. This can also be known as something such as a trophic cascade. A prime example of a trophic cascade in our backyard of the Monterey Bay is that of the sea otter. Throughout the 20th century, the sea otter was rigorously hunted for its pelts, and the sea otter is a natural predator of sea urchins in a kelp forest environment, and sea urchins consume kelp. Therefore, this intense anthropogenic fishery upon the sea otter decreased the predatory pressure upon sea urchins, therefore allowing them to increase in abundance. And with sea urchins increasing, their grazing upon kelp also increased, therefore decimating kelp in a lot of local areas that we see in the kelp forest, creating urchin barrens. And this is important because this is a trophic cascade. This is the result of trophic interactions within a marine food web, but kelp specifically is a foundational species. So in addition to it no longer being a trophic resource, it was also no longer a source of habitat to a lot of different animals in this region, therefore decimating a lot of the biodiversity that we typically see within a kelp forest. Similarly, there's resource competition for trophic resources within a marine food web. For example, here, these would be two individuals belonging to the same species, a whitefish and a blackfish, but within the same species, both consuming crab. Through the consumption of crab, they're able to grow and reproduce and increase in abundance, but therefore this increased abundance would increase the predatory pressure amongst a crab, therefore decreasing its abundance overall, and probably forcing one of these individuals to forage for a new trophic resource. So we would see here that the whitefish would likely be competitive and competing for that resource, still consuming this crab, but this other individual would likely have to turn to something such as a clam or other bivalve in order to perpetuate its abundance throughout its realm. And this sort of specialization that we see here and this variability is what we define as the trophic niche or the dietary niche, which can change throughout one's life cycle. Individual specialization and intertrophic variability is really popular within species of fish that are really highly diverse and biodiverse, such as the rockfish. The rockfish genera is home to 100 different species that both coexist within the same geographic distribution of the northeastern Pacific Ocean. And as a result, this family is able to adaptive radiate and live within the same area, likely because it's both habitating different areas, but as well as foraging for different trophic resources. Which brings us to the idea of the niche variation hypothesis, that populations with wider niches are more variable than populations with narrower niches and tend to be more tolerant to environmental changes. For example, if climate change were to affect the northeastern Pacific Ocean and vastly wipe out invertebrates, there's likely some species of rockfish that would no longer be able to survive because they vastly consume invertebrates for the most part. But there's other species of rockfish that probably consume more fish or other trophic resources that would likely still be able to survive. So since the rockfish family as a whole is very biodiverse and eats a bunch of trophic resources, some sort of environmental change to that system would still allow it to perpetuate. And this degree of individual specialization as well as trophic variability can be impacted by both environmental and biological factors. Environmentally, one of those factors is oceanography and primary productivity. We see with this figure here, relative biomass on the y-axis, as well as nutrients on the x-axis and light limits. And the relative abundance of these organisms such as seagrasses, macroalgae, and phytoplankton can fluctuate depending on nutrients and light and thus primary productivity within a system. This would have sort of a bottom-up effect on the types of primary herbivores and primary carnivores that can coexist in that system due to the presence of seagrasses versus macroalgae versus phytoplankton. Another one of those factors being climate change. We see here the impacts of acidification on a hard-shelled organism under intense acidic waters. Acidification is one of the primary effects of climate change in oceanic waters, and if an organism is to be relying on hard-shelled organisms, it might have to specialize or have more variability towards organisms that don't have hard shells, such as fish or other pyrosomes or gelatinous organisms overall. Habitat can also be a feature that determines trophic variability within a species, something as drastic as a kelp forest versus a coral reef, or just where you're foraging or eating in a water column. So if you're foraging more towards the surface of the ocean or more near the benthos, the drastic difference between kelp forest and coral reef would be more applicable to a species that's highly migratory, so like a tuna or a marlin or a shark are able to coexist in both coral reef and kelp forest environments. Thus, wherever they are at in their movement and migratory pathway is going to determine the types of trophic resources available to them in those areas. Geography has a play in all of these. For species that have very broad or wide geographic distributions, there are going to be fluctuations in both oceanography, primary productivity, climate change effects, as well as habitat that will also determine the sorts of trophic resources available to them. So yes, the environment does have an impact on the trophic variability, but so do biological factors, one of those being gender, specifically species that exhibit sexual dimorphism. A primary example of that would be the anglerfish that we see here, which is our idea of the typical anglerfish that has the lure that attracts certain prey items is what we know as the female part of the species, and the male species is actually parasitic, so it has a completely different trophic and foraging strategy, and it's this small little guy that we see here versus the female here. So gender can very much play a role within a singular species and the different trophic resources that they acquire. Another example would be ontogenetic development. In this figure here, we see brown trout as well as Atlantic cod. As they progress through their life cycle, they are consuming different trophic resources. Typically, as they're getting older, they're eating more complex, higher trophic level prey items. Another primary example of this would be something as a salmon, which is an anadromous fish, which lives in both freshwater. It is born there. It eats freshwater resources until it moves into the marine environment, thus eating more marine resources until it comes back to freshwater to spawn. And again, size and morphology can also be an impact of this, which again can be related to gender or ontogenetic development, or just two individuals that are both sexually mature and of the same size roughly within a species could have different morphologies of their mouth that allows for or not allows for them to be able to have a large enough gape size to eat certain prey items as well. So I've spent a lot of time talking about specialization and trophic variability as it relates to specific prey items, which will be done through my study through stomach content analysis. But another important part of trophic studies is also the isotopic effect of dietary niche, which we would define as the isotopic niche breadth. And this is important to use in conjunction with something like stomach contents, as well as identifying specific prey items that differ trophically, because isotopically, this gives us a longer-term idea of what something is eating and aggregated over time, whereas something as we're just looking at the specific prey items is more of a shorter term look into diet studies. So this is used in conjunction with stomach contents to give us the real understanding of how the dietary niche is varying, both isotopically and stomach content wise. And stable isotope-wise, we look at two different stable isotopes, one of those being carbon-13. Carbon-13 is used as an indicator of variability in primary production pathways, and more negative or greater values of carbon-13 would be indicative of more pelagic and offshore production, whereas something or a less negative or a smaller carbon-13 value would be indicative of neritic or nearshore production. So this figure here shows fish in two different lake environments, but their primary production pathways are vastly different, which drive the overall niche size as well as if they're dietary specialists or generalists. For example, here we have a macrophyte-dominated lake that also has and supports epiphytes. Therefore, the fish within this environment are able to be specialists, and they can have a larger trophic niche size, whereas something that's based more in filamentous green algae can only support an invertebrate sediment-based community and thus supports a population of fish that are diet generalists with a smaller trophic niche size. So in addition to carbon-13, we also look at nitrogen-15. Nitrogen-15 is used as a proxy for trophic level, and that's because it evenly fractionates 3 to 4% with every single increase in trophic level. So overall, larger values of nitrogen-15 are indicative of higher trophic levels. And this schematic is really good at putting both of those together. On the x-axis here, we see the pelagic versus benthic pathway, which would be representative of carbon-13, versus trophic level, which would be indicative of nitrogen-15. And then we see the schematic of an open water sound marine food web as it varies both with carbon pathways and nitrogen pathways. So I've defined both the variability and the trophic niche and dietary niche via stable isotopes and stomach contents, and this is important for us to understand the different resources that are required for them to survive. But this variability can also tell us a lot about pollutant accumulation within an individual and how that may vary based off of their trophic choices. One of those main pollutants being mercury in the marine environment. And mercury is important because it's neurotoxic when consumed in high quantities. It can cause convulsions as well as difficulty with motor skills. It also affects reproduction. It's known to cause birth defects as well as just affect reproduction overall, again, when consumed in high quantities or consumed in high quantities when a female is pregnant. And mercury can fluctuate and increase throughout the marine environment through two ways. One of those being bioaccumulation, which we see is highlighted by the top part of this figure here. Bioaccumulation refers to a single organism and the accumulation of mercury over the course of their lifetime within that single organism, whereas biomagnification is highlighted by the bottom figure here, or the bottom half of the figure here, and biomagnification refers to the transfer of contaminants from lower to higher trophic levels through trophic interactions, essentially. And bioaccumulation and biomagnification occur in the marine environment through the marine mercury cycle. So mercury is sourced into the environment both anthropogenically and naturally through volcanic emissions, hydrothermal activity, mantle degassing, and natural weathering, and anthropogenically through fossil fuel combustion, biomass burning, industrial uses, and mining. And these are inputting elemental, Hg0, and ionic mercury, Hg2, into the atmosphere, or in through the surface ocean through riverine influx and freshwater input into the surface ocean. This elemental and ionic mercury can be deposited through air-sea interactions from the atmosphere into the ocean. And from here, ionic mercury is actually methylated by microbes into methylmercury. And this is important because methylmercury is an organic form of mercury, and it's one of the only forms of mercury that's able to be both bioaccumulated and biomagnified through individuals as whole. And this process, since methylation is performed by microbes and it's a biological process, it tends to be related and shows certain trends through environmental factors and biological factors. So in general, methylation of mercury increases in areas that are highly productive, as well as those that are low in oxygen, because it is an anaerobic pathway. Therefore, in areas of low oxygen, microbes use this as a pathway to generate energy for themselves. And then it also increases in individuals of higher trophic levels. That refers to the law of biomagnification. Those of higher trophic levels are consuming prey of lower trophic levels, thus accumulating all the mercury inside of those lower trophic level organisms, as well as mercury increases in those that are older and of larger sizes, which refers to the laws of bioaccumulation, which increases within the individual itself. So why is this important, and why are we doing this for a species such as canary rockfish? So canary rockfish, their diet has been defined before, but typically in aggregate with other rockfish species. So it isn't allowing us to see the sort of variability within the rockfish family of how they're allocating resources. In addition to this, it hasn't been performed to the distribution, the geographic distribution of my studies. So we encapsulate a lot of the variability that might come with geography. Canary rockfish are distributed from the western Gulf of Alaska all the way down to Baja California, but they're most common in this red highlighted area here from British Columbia all the way into central California. But this entire gray shaded area would be defined as the California current system. And the California current system is home to rocky reefs and kelp forests. It's a very productive and biodiverse environment, and that's for two different reasons. One of them being that it lies on an eastern boundary current. There are 4 eastern boundary currents within the oceanic systems, and all of them are highly productive. And that is due to the fact of the way the currents flow along the coast in conjunction with the spiral of the Earth. It allows for surface waters to be pushed offshore, and this upwelling of deeper, high nutrient, low oxygen water to be brought up to the surface near the coast, allowing for very highly productive and high production systems. In addition to this, it undergoes oscillations, the El Nino Southern Oscillation, which goes through phases of El Nino versus La Nina. And El Nino and La Nina events have different impacts on the western versus eastern parts of the Pacific Ocean, but along California specifically, El Nino events suppress productivity, increase precipitation and temperatures, whereas La Nina events enhance productivity, decrease temperatures and precipitation, and as a result, bring cooler water species in more abundance along the coast, therefore further impacting the trophic availability and sources to an individual at the time. So since this California current system is so highly productive, it's also home to a lot of economically and ecologically valuable natural resources. The rockfish genera as a whole, but also Canary rockfish individually. In 2021, individually, it was landed commercially and recreationally around 900,000 pounds, which was valued at $380,000. And rockfish is one of the more predominant mid-trophic level species in the California current system. We see here over time in tons loaded, rockfish is the white area of the catch, so high amounts of tons being catched, and especially in proportion to these other types of organisms. So Canary rockfish, although it was and still is a very economically and ecologically valuable resource, actually underwent an entire failure. The entire groundfish fishery failed off the US West Coast in the year of 2000, and Canary rockfish specifically were at an all-time population low. The current female spawning stock was actually at 8.7% of its unfished level in the year 2000. So in the year 2003, multiple fisheries management plans were implemented in order to recover this resource, and we'll see over time here, this is depletion as a percentage of unfished spawning output for Canary rockfish, and we see the decline over time, and then after the implementation of these plans, an increase back to this high level. This is representative of biomass of a maximum sustainable yield. So that's the goal of fisheries management, is essentially to get over that BMSY point. So in the year 2015, this fishery was actually determined to be recovered and officially opened again. And this is important as my study, the individuals were taken in 2017 and 2018, so immediately following these years of fishery recovery. In 2015 and 2016 as well, there was extreme El Nino events along the California coastline. So my study takes individuals, again, from 2017 and 2018, immediately following this both anthropogenic pressure of fishing as well as natural fluctuation and pressure of El Nino events along the coast. Which brings me to the 4 objectives of my study. The first one being to characterize the diet and associated factors of gut contents and prey items. My hypotheses being that the diets will consist of teleost, decapods, krill, and other nearshore lower trophic level species. That the diets will fluctuate with larger, female, and sexually mature individuals eating higher trophic level prey items than their counterparts, and that Canary rockfish diets will also vary geographically. My second objective, to utilize stable isotopes, carbon and nitrogen, to determine trophic level and trophic niche, and how these are affected by environmental and biological factors. My hypotheses being that carbon-13 will increase with chlorophyll A and decrease with sea surface temperature, and that nitrogen-15 will increase with sexual maturation, length, weight, and Fulton's K. My third objective, to evaluate total mercury concentrations and how they may vary based on biological and environmental factors. Again, my hypotheses being that mercury will increase with sexual maturation, length, and weight. Mercury will also increase in areas of higher chlorophyll A, thus higher production, and areas closer to shore, but it will be more of a function of biological traits than it will be of the environment. And my last objective, to determine if there are any associations between stomach contents, stable isotope values, and mercury concentrations. My hypotheses being that carbon-13 will display relationships between pelagic and neritic prey items, that nitrogen-15 will vary based off the trophic level and the proportions of those prey items, that mercury will increase with increased presence of higher trophic level prey items in the diet, and that mercury will increase with nitrogen-15 values. So, moving on into the methodology of the study, I wanted to take a minute. I know she's listening. Rachel Brooks, this study would not have been possible with all of her hard work. She is the one who collected all of these fish. She's pictured here. She spent 2 summers of her life and many more years writing her own thesis. She essentially collected enough data to fund 2 whole theses, which is crazy and an amazing effort. So I wanted to take a minute to shout her out here because without her, my study would not have been possible whatsoever. So she went to 13 different ports as pictured here in 5 different fisheries management regions in the summers of 2017 and 2018. She collected 1,500 fish, which is summarized here. There was only 351 that had stomachs that still contain contents. We used another 501 for stable isotope analysis and another 288 of those dorsal muscle tissues for mercury samples. She also recorded a lot of variables about the individual fish before she euthanized them. So she took standard length, weight, sex, and sexual maturation status of the fish, as well as the gonadosomatic index, which is a ratio of gonad weight to body weight. And this is a metric to indicate how much energy is being used for sexual reproduction at the time. Hepatosomatic index, similarly, is a ratio of liver weight to body weight, and this is indicative of the energy reserves left over in a fish. And Fulton's K is a ratio of weight over length cubed, and that is a body mass index of a fish, where a value over 1 is indicative of a more fattier fish, and a value under 1 is a bit of a skinnier fish for its overall size. Environmentally, depth at which the fish were caught was recorded, relief, which is a metric essentially of the complexity of the topography of the seafloor. So 1 being not complex at all, not really a lot of hills, 3 being very complex, very hilly. Latitude, longitude, management region, and port. And in addition to this, I calculated average sea surface temperature as well as average chlorophyll A, and I calculated this using NASA's Giovanna satellite database in their MODIS datasets, which takes 8 measurements a day, and I averaged these from 2008 to 2018 based off the coordinates of where the fish were caught at each individual port to produce average temperature and chlorophyll A values. In order to look at all the stomach content analysis, or all the stomach contents, I took each of the jars and dumped them over a 500 micron sieve that looked sort of like this and allowed for all the ethanol and preservatives to filter through. I then visually inspected and identified everything down to its lowest possible taxonomic level. So for an example here, this is an example of just an ordinary stomach. This is a crab, this would be a teleos, and the rest of all of this would be krill. And so I would count everything for abundance as well as weigh everything to the nearest milligram for weight. And if something looked like this, which a lot of them did, I could identify that this was krill because there was a red tinginess to the tissue. But in order to count for abundance, I had to count all of the individual eyes, all of the black eyes, and have it in abundance, which was really fun. And I also did that for otoliths, even though those were a lot less common. This was probably the more common thing I ever had to deal with. And then in addition to this, I also calculated a variety of prey metrics. I calculated both proportion by number and proportion by weight. I also calculated frequency of occurrence, which essentially tells us how often a prey item is showing up in all of the stomachs. And then prey-specific index of relative importance, which uses both proportion by number, proportion by weight, and frequency of occurrence to indicate the most significant prey items overall to diet. And lastly, I also calculated trophic level, which I use as it's a summation of the proportion of prey items by weight multiplied by a generalized isotopic measurement for that prey item, and a summation of whatever is in the stomach for that organism to produce a trophic level measurement. Mercury analysis was conducted here in the Marine Pollution Studies Lab, and Jessica Heath is here, and she's the one who did all this and helped me out with everything, and she let me entertain her space for a lot of time, so she was very nice for doing that for all of me, or for me, throughout this whole summer, I believe. It was like 5 months of us doing all of this. So essentially what we did is we aliquoted dorsal muscle tissues into little boats. They were inserted into this machine, and within this machine, they're dried and thermochemically decomposed inside of a decomposition furnace, which then moves to the catalyst furnace through flowing oxygen, where oxygen traps all of the excess halogens and oxides within the sample. The imagulator is then rapidly heated to release mercury in its vapor form. And this vapor form of mercury is sent through 2 absorbent cells, and under an atomic absorption spectrophotometer, it measures the quantity of mercury twice at 2 different sensitivities. And these 2 different measurements are calculated using calibration curves and absorbance values to read out a value of parts per million or milligrams per kilograms. And that value is based in total wet weight of total mercury. So total mercury within the sample, not just the organic form. But I used this value and assumed that 95% of this total mercury is organic due to prior research of Bloom 1992. And this is a quicker, cheaper, and fairly still accurate way of assessing organic mercury within a sample without necessarily divulging and separating methyl mercury out. So there's a lot of different methodologies, a lot of different data points. So how do I plan on putting this all together to represent it visually and analytically? For stomach content analysis, I use a permutational multivariate analysis of variance, and that is a permanova. That was just to indicate how stomach contents varied throughout different conglomerations of variables. I also use a distance-based redundancy analysis, or a DBRDA, and this indicated the specific variables that were contributing to variance in stomach content analysis. Further, within the R coding package, I use the stable isotope Bayesian ellipses in R, otherwise known as SIBR, and this allowed me to calculate isotopic niche breadth as well as isotopic measurements of the individual isotopes. I also use a variety of generalized linear models and an AIC. An AIC is essentially you put what you want into a generalized linear models, and it compares different conglomerations of those variables to output the best fit model for that GLM. And the goal of an AIC is to eliminate any variables that display multicollinearity, so variables that represent the same variance within a data set. So it essentially would output the model that has the least amount of variables, but the most amount of variance being represented with those variables. And finally, I also use a principal component analysis, a PCA, and this was just to help to put all of these variables within a singular 2D space and see if there's any linear relationships between stable isotopes, prey contents, and mercury concentrations. So I know that was a lot of jargon and hubbub, but I have results for you as well as they are pictured illustratively. So stomach content-wise, I found 27 total prey items, which I consolidated into 11 higher taxonomic groups. The first being teleos. Anything that I was able to identify down to the species level was through identification of otoliths. Otolith bones in fish essentially are like species markers. They're all individualistic to the fish. In addition, I found shrimp, again, a lot of unidentified, but also the Charonchidae family and 2 specific species that had not really been digested yet, so I was able to identify. Crab, a lot of them looking like this, a lot of unidentified, as well as a lot found in the megalop or development stage, and 1 specific species. I also found squid. The one, again, that I was able to identify was due to the squid beak, and squid beaks are similar to fish otolith bones, that they're unique to the species. Krill, again, a lot of them looking like this, a lot of them just looking like red mush. Pyrosomes, a lot of them looking like this as well. Amphipods, isopods, copepods, unknown decapod. Unknown decapod occurred when I found different crab-like claws, that's the word for it, claws, or like segmented body parts that I couldn't necessarily identify as a crab or a lobster or a krill, but I knew it fell under the decapod prey group. And I also had an other category. Unidentified tissue occurred when I had different prey items within a stomach, and then I would have to, if this tissue was just standing there, but I couldn't identify it to a specific one of those prey items. Parasites, and then I also found a singular piece of algae, which was really interesting. But this is all summarized here. The total number of stomachs within these prey groups are found, total abundance, proportion by number, total weight of the prey item, proportion by weight, frequency of occurrence, again, how often something's occurring, and PSIRI, which is a conglomeration of all those variables to tell us the most significant prey items within the diet. We see that teleos had the highest weight as well as proportion by weight. Also see that krill are the highest in abundance and proportion by number. And overall, we see that krill is the most frequently occurring, and then crab. So even though crab wasn't high in number or weight, it was really frequently occurring, which is an important indicator of what something is eating overall. And then teleos. But then overall, the PSIRI indicated that krill and teleos were the most significant prey items using all of these metrics. This is just a representation of how percent by weight and percent by number, weight is red, number is blue, and how drastically that can look when looking at stomach contents and why it's important to use both weight and number. They can look very different, but they do tell us both important things about stomach contents, and that's why they must be used in conjunction with each other when doing a stomach content analysis or a trophic ecology study. So summarizing, krill is the most abundant, teleos is the most important by weight. Both appear really frequently in the stomach samples, with crab actually appearing the second most often. And then teleos and krill are the most important, relative, significant overall prey items within the diet. So I also wanted to look at how this is varying geographically. So this is the result of my permanova. The axis are NMDS1 and 2, which stands for non-metric multidimensional scaling space. And these are 95% confidence intervals of each of the management regions with all the different prey items and how they vary within each other in the management regions. Important things to see here is that the Vancouver ellipse, the most northern ellipse of latitude, orients itself differently than the other ellipse. It orients itself more so with NMDS2 than NMDS1, indicating that it's more different than all the other management regions. In addition to this, we also see that squid and decapod are driving a lot of the differences as seen within the management regions. And when we break this up as a stacked bar plot within all the regions, again, all the regions are ordered from north to south. We see that decapod prey group is really prevalent in the Vancouver region, and the squid prey group is really prevalent in the Conception region, but not really present or there at all, really, in all these other management regions. And an explanation as to why that Vancouver ellipse might be orienting itself so differently is that it also appears to be the most proportional in terms of composition of prey items. All of these other management regions seem to appear, or they seem to specialize in one or two prey items specifically, whereas the Vancouver region has a really even distribution of all the prey items, maybe insinuating why it's orienting itself differently in NMDS space. For example, in Eureka and Conception, they have a really high proportion of krill, and in Columbia and Conception, they have a really high proportions of teleosts. In addition to this, I also broke this up by port, again, ordered from north to south. And what we'll see here is that there's a lot more finer scale variability when looking at prey composition at the port level versus the management region level. We'll also see that krill and teleost prey items are the only prey items that are present at every single port, and that even ports that are right next to each other can vary drastically in terms of prey items, but then when we zoom out and get a larger geographic look at the data, it seems to be more even in terms of proportion as opposed to looking at the port level. So I also wanted to see if there were specific variables that were driving this. Through the DBRDA, I found that chlorophyll A, depth, longitude, GSI, and relief were all factors that were contributing to differences in prey items, which is represented here in cap scale space, which is similar to NMDS1. It's just like a non-metric way of representing data. And we have variables here on the left and prey items on the right, and in which these variables point or these prey items point would indicate that that is a driver of dissimilarity within these management regions which are plotted. So for example, crab in the Columbia region, pyrosomes in the Conception region, and then krill in the Eureka and Vancouver regions are likely causing a lot of dissimilarities between individuals in those regions. So moving on to stable isotopes, this is again just a summary table of all the raw data that I took. So carbon-13 as well as nitrogen-15 in mean trophic level, which is also represented here in isotopic space. We have carbon-13 on the X, nitrogen-15 on the Y, and again, just that relationship of how these vary, so trophic level with nitrogen and pelagic versus benthic with carbon. But we don't necessarily see any sort of linear or relationship here when we're looking at it all together. So when we break it down, carbon-13 here, we see a more distinct relationship. We see a decrease in carbon-13 with depth. But when we look at nitrogen-15, it appears to fluctuate back and forth, not really displaying like an environmental or latitudinal trend with nitrogen. And that's likely because carbon-13 is a function of primary productivity, so it likely is going to have some sort of environmental or latitudinal effect, whereas nitrogen-15 itself is supposed to vary more biologically. This is also represented by ellipses. These dotted lines are convex holes, so that just represents all of the actual individual points within a region, and the ellipses are 95% confidence interval ellipses of the points within the management regions, which is also broken up by individual region. A wider ellipse would indicate individuals in that region are foraging in a plethora of primary productivity pathways, whereas a longer ellipse would indicate individuals in that area are varying in terms of trophic level. What we see is that the Columbia region has the largest carbon and nitrogen values, as well as Vancouver has the smallest carbon and nitrogen values, and those two regions lie directly next to each other in the geographic spread of my data, which is interesting. So then I also wanted to see if there were specific variables that were driving all this variability in these specific isotopic measurements. So I ran GLMs for carbon, and what I found is a lot of variables were contributing to variance in carbon. Carbon was negatively related to depth, maturity, and HSI, and it was positively related to relief, chlorophyll A, sea surface temperature, and latitude. And what we'll see here is just a representation of all those negative relationships. So we see fish that are caught at deeper depths, as well as fish that are sexually mature and weigh more, tend to have more negative values of carbon-13, which would be indicative of fish that are eating more pelagic pathways of prey items. And those caught at shallower depths that are sexually immature or weigh less are foraging in more benthic or neritic pathways. Similarly, for nitrogen, I ran a GLM. It was positively related to chlorophyll A, sea surface temperature, and latitude, which is represented here. Chlorophyll A on the left, sea surface temperature on the right. And what we'll see is port is also aggregated from north to south. So although there are these positive relationships with chlorophyll A and sea surface temperature with nitrogen-15, it doesn't follow a latitudinal trend. You'll see Nia Bay is the most northernmost place, and it's like in the middle of productivity. It's not on either end, as you would think with a latitudinal spread. In addition to this, temperature almost varies like an entire degree when you look at these more southern ports of Half Moon Bay to Santa Barbara versus Bodega Bay to Nia Bay. There's like this distinct divide in temperature, which would likely mean that the Southern California countercurrent and these warmer waters are having more of an effect in these areas than these more northern latitudes, which could be home to supporting different species and food webs overall, therefore contributing to dietary niche variation, which is known to happen in the California current system. It's been documented before that there are a lot of fluctuations in both temperature as well as chlorophyll A. This is just looking at California specifically, but this is looking at California, or the California current system as a whole. You'll see that temperature is a bit more consistent with latitude, but chlorophyll A definitely does have high fluctuation and doesn't follow some sort of trend along the coast, both within California as well as along the California coast in general. Moving into mercury concentrations, again, just another summary table of all the raw data. I found that mercury was highest in the Vancouver, the most northernmost area, and the variability was also really high, versus in the southernmost region was the lowest values of mercury and the lowest and some of the lower variability, which is again represented here. These black lines within the boxes are median values, not mean values, but we would see overall there is a slight decrease in mercury concentrations from going from north to south. There's also a decrease in variability from going from north to south. So again, wanting to see if there's any specific variables that are driving this. And what I found is that sex, weight, Fulton's K, and longitude are significantly related to mercury concentration values. And since weight and Fulton's K are made up of weight and length, I plotted these here. Weight on the left and standard length on the right with mercury on the y-axis. And we'll see strong positive, moderately strong positive correlations between mercury and length and mercury and weight. But Fulton's K, as we know, was negatively related, which is interesting because it's made up of weight and length. But the negative relationship with Fulton's K would suggest that those that have high mercury concentrations tend to be really skinny, have a lower body mass index. And that makes sense as you have less tissue on your body, you're not biodiluting the mercury as much, therefore having higher concentrations of mercury overall. I also wanted to see if there's a difference in terms of sexual maturation, because when I was running just different calculations, I found that it seemed to be a pretty drastic change between immature and mature individuals, which is plotted here. I ran a separate Welch's 2-sample t-test to indicate if this was significant, and it was. So those that are sexually mature tend to have higher average mercury concentrations compared to sexually immature individuals. So in order to tie together these three seemingly separate studies of prey content, stable isotopes, and mercury, I decided to run a synthesis of generalized linear models. I did mercury in prey items, mercury in isotopes, and then all of the individual isotopic measurements against prey items. And I found some relation with mercury. Mercury tended to increase in individuals that ate a lot more krill and a lot more teleos. Mercury also seemed to increase in individuals that had higher trophic level, had higher nitrogen values. And mercury also seemed to be higher in those that were foraging in the pelagic realm, so with lower or more negative carbon-13 values. Carbon-13 had relationships to shrimp, crab, and krill, but it fluctuated back and forth, therefore not really showing like a linear trend within high, low, and moderate concentrations of, or values of carbon-13. Similarly, nitrogen-15 wasn't really related to things with teleos, but nitrogen-15 tend to increase in individuals that ate less krill, and trophic level overall increased in individuals that were eating higher amounts of teleosts. So that was a lot of like just words, so I wanted to see if I could represent this visually within a 2D space, so I ran these principal component analysis, and all of my PCAs are colored. The dots are colored by mercury concentrations, so low being green, moderate being red, and high being blue. And in this PCA, I inputted stable isotope and prey item data, and what I found is that the high ellipse is a bit more separated from the low and moderate ellipse. That high mercury is being driven by increased teleos presence in the diet and higher nitrogen-15 values, and that lower mercury is being driven by increased crab, shrimp, and copepod presence, which is related to less negative carbon-13 values, which is indicative of neritic or benthic nearshore production. Again, I ran another PCA colored the same way with mercury, but based off of biological values. And what we see here is that high mercury is being driven by increased allocation towards sexual reproduction, as well as those that are way more and are longer. And then environmentally wise, this didn't seem to be a good indicator of mercury concentrations, as there's a lot of overlaps of the ellipses of mercury. But what we did see is that mercury was being driven by increased presence of chlorophyll A, deeper depths, and those of lower relief environments. That's a lot of information. How does that all relate? Why is it important? And what can we draw from this whole thing? Krill was definitely the highest in abundance, and teleos the largest in weight, and those look very differently when you look at a stomach content. So it's really important to be using both abundance and weight when you're doing stomach content analysis, because it can tell you two very different things about a diet, and without one or the other, you would probably have a bias in your study. I also found 27 prey items, but it was vastly dominated by 2 different prey groups, indicating that canary rockfish are likely specialists within the California current system. And prey proportions were influenced heavily by environmental factors, not really biological factors. So geographic distribution was really what was driving differences in diet, and squid and decapod prey groups of that is what's driving most dissimilarity amongst regions. We saw in these more northern latitudes that there's increased reliance of crab and shrimp and decapods, so more of like a neritic and benthic foraging area, whereas in these more southern regions, we see increased presence of krill and teleos. We also see an increased presence of squid. Squid is likely there because there's a high commercial fishery for squid in the Southern California region and not really in these other areas. So prey proportions are definitely being driven by geographic distribution. Stable isotopes seem to vary a lot as well. We found a 3.7 per mil range in carbon-13. And a negative association would be indicative, again, pelagic offshore, positive association, this neritic and benthic production. So carbon was related, so those that were caught at deeper depths and that were sexually mature and had higher energy reserves were likely foraging in the pelagic and offshore environment, versus those that lived in higher reliefs, higher productivity, higher temperatures, and more northern latitudes were foraging in the more neritic and benthic area of production. And although we see these relationships with isotopes and the variables, there was no direct correlation with prey types, again, further highlighting why we must use both isotopes and stomach contents when looking at the entire dietary niche. Nitrogen-15 was related positively to temperature, productivity, and latitude, which is interesting. You wouldn't think that temperature, productivity, and latitude all positively correlate with one another. Nitrogen-15 was also higher, and thus trophic level higher in those that were eating less krill. And this weird association between temperature, productivity, and latitude might be a function of the 2015 El Nino that preceded the collection of my events, and that's because El Nino can drastically alter the nitrate availability within the water column, and the different Prochlorococcus versus Syncococcus and the different cyanobacteria that are evident and prevalent throughout the system that changes throughout the pre, during, and post El Nino, which could be impacting the nitrate utilization at the base of the food web and thus impacting the stable isotopes that we see in my samples after the El Nino. Trophic level was related to temperature and length, and so I made this little schematic here to maybe emphasize why that might be. With increasing length, we see an increase in metabolic requirements and maybe even a diet shift to help satiate these new metabolic rates in these newer sizes. So individuals within the same species that are smaller might be foraging smaller prey items, but as they get bigger and have bigger metabolic requirements, they might have to shift to something more dense, such as a fish. This can also be impacted by something like temperature. Temperature has been known to cause metabolic stress within an organism and increase the metabolic requirements of an organism, thus maybe partitioning and forcing this shift to a higher trophic level prey item sooner on. Mercury was also related to mostly biological factors, length, weight, maturity, and Fulton's K. And we can see as we look at different fish within the marine environment, these lower mercury content fish tend to be our forage fish, so anchovies, sardines, these smaller fish that don't live as long. And as you get higher and higher throughout these mercury concentrations, we see our marlin, our tuna, our sharks, our swordfish. Those are much larger individuals. They also tend to live a bit longer, thus accumulating more mercury over time. That's why it's so much related to biological factors as opposed to environmental factors. However, it's also important to realize that proximity to freshwater inputs could also be increasing mercury concentrations. I put stars throughout my system that indicate these river runoff sites, so the Puget Sound, the Columbia River, San Francisco Bay, and the Elkhorn Slough, even in Monterey Bay. These are just some examples of freshwater inputs along the coast. And with that idea of coastal upwelling that we see within the California current system, in addition to this input of mercury from freshwater sources, could definitely be conducive to hotspots of mercury along the coast. In fisheries management as a whole, fisheries management plans tend to conserve smaller or sexually immature fish from commercial or recreational take to allow populations to reproduce. But as a function of this, it's also allowing for these larger fish that have higher mercury concentrations to be consumed. We even see that larger fish in general are more likely to be fished and consumed. Here is the 2018 most popular marine seafood. We see things such as tuna, pollock, cod, and even salmon. And all of these things can be conducive to higher mercury concentrations, and they're also the most sought after and fished for food. And since they tend to be bigger and live longer, they could be the ones that have higher mercury as well. This is important to look at even locally as well. This is a figure from the Humboldt Baykeeper, which just advertises the best, good, moderate, and avoiding fish choices when it comes to mercury. And we can see that Canary Rockfish is actually in the in moderation serving, but when you look at rockfish as a whole, it spans this entire pictograph. We have black rockfish in the good choices, Canary rockfish in moderation, and then we have some that we should just be avoiding altogether. So again, further highlighting that research need to indicate mercury concentrations at the species level, not only at the family level. The FDA actually has a legal action limit for seafood. It's at 1.0 parts per million, but the state of California actually advises against the consumption of fish with a 0.5 part per million of mercury. And individuals in this study, as well as in Davis et al., are finding fish along the California current system that are approaching these levels or already at these levels, and they're only mid-trophic level fish. They're not even these apex predators that we're known to be consuming in high quantities, which is shown by this figure here, all the way from Alaska and coastal fish along Washington through California. Further research into the regional hotspots of mercury. This is again another figure from this Davis et al. Paper. We see different hotspots along the coast, especially near areas of urban development like Los Angeles, or freshwater inputs or more urban development like the San Francisco Bay Area tend to be home of high mercury concentrations. In addition to this, we see just species of interest as well. This is an aggregate by fish family and this high or the fluctuating amounts of mercury concentrations. So further research into species of interest as well that are containing these high concentrations of mercury. But from my study, can we estimate canary rockfish with higher mercury concentrations and hopefully put in fisheries management plans that would likely deter from the fishing of high mercury individuals? And we found that those that are eating more pelagic items like Atelios and less of those benthic production items like shrimp and crab are conducive to higher mercury concentrations. Those, again, with higher nitrogen values, maybe shifting fishing away from these northern latitudes or even just fishing away from more productive environments. Maybe even putting a depth cap in the fishery, so no longer catching fish below a certain depth could help to mitigate the concentrations of mercury in this fishery. As well as there's a length and weight restriction in terms of catching with normal fisheries management, maybe putting a length and weight cap that you can't catch and sell fish of a certain length and weight would help to mitigate this problem as well in the fishery. So the main takeaways from all of this study being that prey contents are impacted by both short-term changes to foraging strategy and the environment, which over time can influence isotopic values and reveal long-term behavioral shifts or shifts in biological traits. that canary rockfish appear to be generalists, but in reality are a heterogeneous compilation of specialists impacted by productivity, temperature, location, and ontogenetic development. That the protection of krill and other teleost populations are just as important to the perpetuation of the canary rockfish stock as the protection of canary rockfish itself. And that the increased presence of mercury in mid-trophic level species is being impacted by ontogenic development and geographic location. With that, that is the end of my defense, and I'll take any questions or comments. Yes.
So like from site to site and like in the different regions you saw a lot of variation. Like between individuals that you were looking at, what was the variation between them, or was it pretty like standard between?
For mercury or for stomach contents?
For gut content.
Gut contents, I think it was really just based off of that geographic location. Like when I looked at biological variables, like I performed the Permanova example for like different genders and for different like sexually immature and immature fish, and the ellipses just like had a lot of overlap. Like they did not vary a lot, like they didn't show a lot of like different alignment with the axes. It seemed to be really based off of geographic location, and especially when broken down at the port level, that's where it seemed to have a lot of high variability. But when you looked at like ports all conglomerated in a region, it didn't seem to have as much variability, if that makes sense. So I think it's really dependent on like fine scale distribution of trophic resources within a singular port is driving the difference we see. Yes.
Well, I guess first off, thanks for your talk. Wonderful. I guess following off of that question, the variability versus in the ports versus the regions. I know you mentioned that it's interesting that in the Columbia regions versus the Vancouver regions, you had kind of a greater, you had kind of that maximum distribution in your nitrogen, isotopic nitrogen, isotopic carbon concentrations versus your Columbia. Forgive me if you mentioned in your talk where you thought that might, why that variability might exist, but what were you thinking as far as why you were seeing those?
I think there could be multiple reasons. The program itself is supposed to code out for this, but it could still possibly be driving the difference that we see is that the Vancouver region had the lowest amount of samples and Columbia had the highest amount of samples. And the program in R is supposed to account for that, so it's not supposed to be driving that variability. But I could also suggest that like individuals within those like regions, like I think it was Vancouver was more similar, it had less of a range, that those individuals are probably all like one type of organism, like they're all sexually mature, or they're all like sexually immature, or they all have similar like length and weight values, versus something like in Columbia that had a lot of high scale like variability. There's individuals from every walk of life, every development stage, every length and weight size, and maybe that's why we see more of a variability there.
Okay, thank you.
Mm-hmm.
This is more of a hypothetical, but kind of along the same line of questioning. Do you think a life stage might impact it? Like, for example, we saw a lot of megalops, or like if they were consuming older crabs, might there be more mercury in those crabs?
I think so, definitely. I hadn't really thought about that, but yes, I do think so. I think it'd be interesting to see if you can do a study kind of based off that, just because it's hard when you're looking at contents to see like where they really are in terms of maturation, because a lot of the time they're partially digested. But that would be interesting to see if like you could indicate their level of like where they're at in their life cycle and seeing if that impacts isotope values overall. Yeah.
Did you see any indication of historical mercury trends over time in rockfish and equally? So if you have juveniles that are on a lower mercury concentration than are alcohols versus juveniles on a higher mercury concentration, do you see any indications of that, or different concentrations not change on that?
I never looked into that. I think that's actually a really good point, though. I think I probably could have done more work with Rachel's data set because she did a life history study of the rockfish. So maybe using their age and somehow using that to indicate mercury concentrations and how that is related through the different stages of the fish could have been interesting. I think I could have done a lot more with Rachel's like data and stuff. I just felt like I had a lot already here. And so I was like, I don't know if I want to bring that whole thing in the mix, but maybe Rachel and I can do that later. Maybe it'll be like a future paper. So that's a good question. I should look into it.
At one point, you showed us like the different levels of like fish to avoid versus fish to eat in moderation, and it showed the Canary Rockfish and fish to eat in moderation, and you sounded like a little bit surprised when that was there. Especially like I assume you look at this fish because the mercury levels are higher. So based on the levels that you were seeing, was it how did it compare to, I guess, the levels that they're basing that scale off of? Like, did you feel like it should be in something that you should avoid, or?
I think based off the conclusions of my study, because I found up to like 0.5 part per million, and that's like right at the state of California's like cap of like seafood consumption. I guess it surprised me because you think of rockfish as maybe something that is like a lower trophic level species. It's not really high up there. Like you don't really think that it's gonna be accumulating a lot of mercury, but because it is long lived, that might be more sort of the issue here. But those ones that were like in the red category, those tend to be a lot bigger. So like our vermilions and things like that, those tend to be a bit bigger. So that makes sense as to why I was there. I guess going into this, I didn't realize that it was going to be as more like a persistent of an issue as it was. And so I was surprised to have found it that high, but I'm glad that I did the research to find out.
Yeah, I can answer. I was just curious to know, so like the fish that Rachel collected was in 2017, so there's 2 years of the fisheries being open.
Mm-hmm.
Do you think that potentially had an impact or influenced your results in terms of gut contents? Like if an area was more intensely fished than another, example? Yeah.
I think, yes, that could definitely have an impact, but what I hoped for that would be controlled for in this experiment is the fact that they were both fished for in the summer for the most part, and normally a lot of trophic habits can change throughout the year, but typically when you're looking from summer to summer, they won't hopefully change as much. I mean, they definitely can because it's a whole different year, but typically if you can just fish within the same season, it's a hopes that they'll be eating similar diets. But yeah, the year-to-year variation could definitely be an impact or a bias in the study for sure.
Questions submitted online here by Greg I.A.
Oh gosh, okay.
The matter-rich organs of Canary Rockfish were measured for mercury, perhaps higher in the liver than the muscle. Also, in what organs would mercury bioaccumulate or bioaggregate? And it says in sable fish, mercury concentrates in the fatty tissues like the liver is higher in hemoglobin than other specimens. But of course, you can usually eat fish muscle also doesn't matter. So.
Yeah, I mean, it can definitely vary, both isotopically and mercury measurements can vary based off of the turnover rate of the tissue. So both the turnover rate can impact the isotopic values I measured as well as the mercury values. But yeah, I think he made a good point that it's probably better for me to be measuring the actual, because I use dorsal muscle tissue. So using muscle would probably be the most accurate or most, I guess, prevalent to women or human health than anything. But yeah, I think that it does. I know that inorganic mercury accumulates in cell membranes, and that's why it's not able to be passed along from fish to fish, is that's excreted, and that organic mercury is kept in the cytoplasm, and that's why it's able to be biomagnified. So I would assume that maybe that mercury can be held differently in different muscle tissues as well, considering just the species of mercury determines where it's held in the cell as well. So yes, I do think that could have an impact.
All right, any final questions? Otherwise, the committee will take her